Welcome to the seminar. My name is Lauren Updike. I'm the director for the Whole You and super excited to be with Dr. Kawasumi today. So I'll give maybe 2 minutes. And the session is recorded, so we will get this seminar on our YouTube channel. Hey, Chris. Awesome. So yeah, this great seminar will be on our YouTube channel probably in the next week or so. But thank you for spending your lunch hour with the Whole You and Dr. Kawasumi. Hey, Cynthia. Tea drinker, okay. All right, people are starting to jump on. Hey, Brenda. Oh, awesome. Harborview's here. Hi, Lisa. Food system, nutrition and health. Awesome. Hey, Judith. Nurse from UW-MC. Awesome. Thanks for everything you do. Yes, if you're just joining, let us know who you are, where you work. And we'll give 1 more minute. I'm sure we'll have some people kind of transitioning from meeting to their lunch break. So. Hi, Sarah. Hey, Becky. Hi, Caroline. UW-HR. Yay. All right. Well, we're going to go ahead and get started. Feel free to continue to engage in our chat. Welcome. My name is Lauren Updike. I'm the director for the Whole You program, and we always have some really amazing faculty at University of Washington. So we are super grateful for Dr. Kawasumi to join us today. He is the assistant professor at medicine and dermatology. And we are super excited to hear about skin cancer prevention and caffeine. And I will be monitoring the chat. So if you have any questions, we're going to wait until the end of the presentation. So feel free to add them. And then when there's time to go through the questions, I will go ahead and do my best to capture all of the questions so you can get your answers. But welcome, everybody, and thank you again for your time. Take it away. I will go on mute.
Thank you very much, Lauren. My name is Masaoki Kawasumi. I'm a skin cancer researcher in dermatology. So today, I would like to talk about the skin cancer prevention by caffeine, another reason to love coffee. My research lab is located at South Lake Union. I took this photo on July 4th several years ago from one of our research buildings. So you can see many yachts on Lake Union. Probably people enjoy sunshine, and maybe some people enjoy coffee. So I'd like to connect those 2 things in this session, sunshine and coffee. So we would like to use a poll everywhere during this session. So please scan this QR code and log in with your UW email address. So I am going to also put the link into chat so you can click and then log in with your UW address because this system is limited to UW users. So here's our first question. So where are you originally from? So you can click the map to indicate where are you originally from. So I'm originally from Japan, so I put 1 point on the map. Thank you very much for your response. So this is great. So we are a group of diverse people coming from many different countries. So some people may enjoy coffee and tea and different kinds of caffeine containing drinks and snacks. Thank you very much for joining this poll. So let me move on to the next slide. Thank you very much. So many people enjoy sunshine, although many people may understand excessive sunlight exposure may cause a skin problem. This may be because sunshine can be addictive. UV irradiated skin produces the endogenous opioid beta endorphin that initiates opioid-like effects as pain relief and dependency. So simply speaking, people feel good under the sun. So that's why people want to enjoy sunshine. So people's behavior may be controlled by rays from the sun like this mannequin. So it may be difficult to change people's sun-seeking behavior. But too much fun in the sun, ultraviolet light from the sun can reach the ground and penetrate skin and damage DNA. And DNA is an important molecule that carries genetic information and that define our body and function. So DNA damage is a big issue. So do you know how many number of DNA lesions can be generated by 1 hour of sunlight? So DNA lesion is a DNA damage site. So DNA damage site can be DNA breaks or some modification of DNA. So my question is that how many DNA lesions per cell are generated by 1 hour of sunlight? So you can join this poll. One, 100, or 1,000, or 100,000. How many DNA lesions or DNA damaged sites can be created by 1 hour of sunlight? So thank you for joining this poll. So you can, if you have a web browser, you can type the link pollev.com/Masaoki-Kawasumi 544. The link is shown at the top of this slide. Thank you very much. So many people choose 1,000. Then you may be surprised by seeing this number. So actually, 1 hour of sunlight generates 100,000 DNA lesions per cell, which are potentially mutagenic. And as UV damages skin, causing aging, sunburn, and skin cancer. So this is a famous picture published in a top journal, New England Journal of Medicine. And you may notice one side of his face is different from the other side. And why? So hint is that he had driven a delivery truck for 28 years. So what does it mean? So ultraviolet A, so this is one type of UV rays transmit through window glass, penetrating the epidermis and upper layers of dermis. So this is the effect of UV coming from the sun. So this is called unilateral dermatohelioses. Here's a table showing 10 leading cancer types in the US. So what is number 1 in males and females in cancer new cases? So this table showed the 10 leading cancers. So what is number 1 in males and females? So the answer is prostate cancer in males and breast cancer in females. So collectively, total number of cancer new cases is 1.7 million in the US annually. And that may lead to 0.6 million cancer deaths annually. But if you look at this figure legend very carefully, cases exclude basal cell and the squamous cell skin cancers. So skin cancers are not included. And in non-melanoma skin cancer, actually the number one cancer cases. So total number is 5.4 million cases annually in the US. So that exceeds all other cancers combined, 1.7 million. So non-melanoma skin cancers are so many and often not tracked by cancer registry. So that's why non-melanoma skin cancer are not shown in these 10 leading cancer types, but actually skin cancer is the most prevalent cancer in the US. So this is the most common cancer, and it's frequently fair-skinned, sun-sensitive whites, and lifetime prevalence is 1 in 5 in the US. And non-melanoma skin cancer mostly caused by long-term UV exposure. And there are two types of non-melanoma skin cancer. One is a basal cell carcinoma. The other one is a squamous cell carcinoma. And melanoma is another type of skin cancer. And melanoma represents less than 5% of skin cancer, but results in most deaths. So this is a more aggressive type of skin cancer. It arises following the transformation of melanocytes, which are pigment-producing cells. So most of melanoma are pigmented, like these images. This is important to remember, the ABCDEs of melanoma, to distinguish benign tumor from malignant tumor. So malignant tumors are asymmetry, and borders are unclear, and a variety of colors, and larger diameter, and it's evolving. So if you see these skin changes, go to dermatology clinic. So here's another question. So which countries have a high skin cancer death rate? So you can select multiple countries. Do you know which country have a high skin cancer death rates? Thank you for joining this poll. So many people chose United States, Canada, Europe, Australia. This is great. Thank you for joining this poll. So I'm going to look at the result. Okay, great. So this is a world map showing the skin cancer death rate. Red indicates high skin cancer death rates. So as many of you noticed that New Zealand and Australia have high skin cancer death rate, and also North America, Europe, and Russia. So these countries are where more fair-skinned people live. Because melanin protects the skin from UV damage. So this is an important slide showing melanin content and UV-induced DNA lesions. Upper panel showed the melanin content in Caucasian skin, Asian skin, and Black skin. And lower panel showed the amount of UV-induced DNA lesions as shown as green signals. So more melanin content in Black skin and less lesions are generated. In fair skin, more lesions are generated. So melanin actually protects the skin from UV damage. So skin cancer is less prevalent in people of color, so 70 times less prevalent in people of color compared to white population. However, when skin cancer occurs in non-whites, it often presents at a more advanced stage, and thus the prognosis worse. So what does this mean? So this slide showed a comparison of non-Hispanic Black population and non-Hispanic white population, showing what the stage of melanoma is found at the time of diagnosis. So regional and distant are considered as advanced stage of melanoma, and a more advanced stage of melanoma is found in Black population rather than in white population at the time of diagnosis. So this is health disparity, so preventable differences. This could be due to healthcare access issue or misdiagnosis or other socioeconomic factors. But how about the science part? So I'd like to talk about a little bit science related to UV-induced DNA damage. How do cells respond to UV? UV generates DNA lesion by forming a dimer at the two adjacent thymines. So DNA is an important molecule that carry genetic mutation, and 4 letters are used, A, T, C, and G. And when T and T are adjacent in DNA, dimer may be formed following UV radiation. So that is called the thymine dimer. So after DNA damage or DNA lesions are generated, many biological processes happen. One important biological process is ATR check1 signaling pathway to sense UV-induced damage and regulate the cell cycle so that the cells have an appropriate time to repair the DNA damage. Second important biological process is called nucleotide excision repair. This is a DNA repair mechanism, and UV-induced DNA lesions are recognized and excised and then repaired in an error-free manner. So this is an efficient DNA repair mechanism, and many studies are done to characterize how this mechanism works. And the third important biological process is trans-lesion synthesis. So this is more complicated, but the UV-induced DNA lesions are highly DNA distorting lesions, so that will block regular DNA polymerase during the cell cycle. So cells cannot proliferate when DNA polymerase is stored at the lesion. So in that case, specialized lesion bypass polymerase needs to be recruited, but this polymerase is error-prone, and the wrong nucleotides are often inserted opposite the lesion. So this is a mutagenic process, and this is how genetic mutations are created after UV. So these are called DNA damage responses. I talked about the UV-induced DNA damage, but the DNA can be damaged by many other factors like environmental factors and chemotherapeutic agents. So ionizing radiation or cosmic radiation damage DNA by inducing DNA breaks, and also chemotherapeutic agents damage DNA, especially in cancer cells. So that's why cancer cells are killed by chemotherapeutic agents. But at the same time, some normal cells are affected by these DNA damaging agents. So we need to think about how to prevent normal cells from dying. Here's another poll. So which one generates the highest number of DNA lesions per cell? So DNA lesion could be DNA breaks or modification of DNA. So how many DNA damage sites per cell by chest X-ray twice, or airline travel 7 hours, or space mission 1 hour? Very interesting to see this response. So thank you for joining this poll. So chest X-ray twice, airline travel 7 hours, and space mission 1 hour. So which one generates the highest number of DNA lesions per cell? Okay, then I'm going to next slide. So this table will give the answer. So let me explain this table a little bit. So this table showing DNA lesions generated by endogenous and exogenous DNA damage. So UV radiation generates the highest number of lesions per cell, but other environmental factors also generate DNA lesion, although different types of DNA lesions are generated. So chest X-ray, 0.0008 lesions per cell, inducing a DNA double-strand breaks, and airline travel, 0.0002 per hour, and space mission 60 days, 2 lesions per cell. So when I calculate to match with my question, so chest X-ray twice, 0.0016 lesions per cell, airline travel 7 hours, 0.0014, and space mission 1 hour, 0.0014. So first one is the highest number. But you can notice that these three are similar number, and this is a very low number. So medical radiation like X-ray or airline travel have a negligible effect on human health. But if you are exposed to a lot of high energy radiation, then that is a problem and may increase skin cancer, not only skin cancer, but also any types of cancer risk. Going back to UV-induced skin carcinogenesis, this is a long-term process starting from a generation of UV-induced lesions. Genetic mutations are accumulating after chronic UV radiation, and then eventually leading to dysfunctional tumor suppressor. So P53 is a famous tumor suppressor protein that will inhibit the development of cancer. When P53 is mutated, that becomes dysfunctional and cancer will develop. So this is a long-term process, and one study estimates that 70,000 hours of sunlight is required for developing a skin cancer. So why does it take so long? One reason is UV-induced DNA lesions are mostly removed by nucleotide exogenous repair. So we have a very nice DNA repair mechanism in human cells. Xeroderma pigmentosa is a rare autosomal recessive disorder of DNA repair. So their cells have a defective DNA repair mechanism. So their cells cannot repair UV-induced DNA damage. So that's why they have an increased risk of skin cancer development. So 10,000-fold increased risk. Lower graph showed the age distribution of skin cancer patients among the general population versus XP patients. In general public, 67 years old for skin cancer development, but 9 years old among the XP patients. So significantly increased risk of skin cancer after sunlight exposure in XP patients. Another reason why it takes so long to cause skin cancer is that both alleles of tumor suppressor gene needs to be inactive. So human cells are diploid cells. So that means we have 2 copies of each gene. Even if one gene becomes defective, we have another gene. This concept is more obvious in elephants because elephants do not develop cancer. So this is an interesting study comparing many different species, and the y-axis indicates cancer incidence and then x-axis indicates body mass and lifespan. So when body mass is big, likelihood of developing cancer will increase, and when lifespan is long, also increase the risk of developing cancer. And the elephant is a good example for large body mass and a long lifespan, but they do not develop cancer. And why? Because elephant has extra copy of a key tumor suppressor. So TP53, a gene encoding P53, humans have 2 copies, but elephants has 40 copies. So they have a more extra system to protect their cells from developing cancers. Third reason could be the multiple genetic hit model of cancer. So this says one mutation is not enough to cause cancer. Many mutations may be required for to become a cancer. This is an interesting study showing ultra deep sequencing of small biopsies from aged sun-exposed normal human eyelid. This image shows the size and distribution of mutant clones in eyelid, and surprisingly, 25% of normal skin cells have a cancer-causing mutations, while it looks normal. So normal appearing skin have many mutations, but they're still not cancer cells. So to make normal cells malignant, many mutations are required. So UV-induced skin carcinogenesis is a long-term process, and the key question is how can skin cancer be prevented? So I'd like to ask you your opinion. I think that this may be, So how can skin cancer be prevented? You can type 1 word or 2 words or 3 words. Thank you for submitting your idea how to prevent the skin cancer. Right, it's nice to see everyone's response. So many people said the sunscreen and clothing, hat, and shirts. These are great questions, great answers. So thank you very much for your response. Yes, so as you said, sun avoidance and sunscreens are great. So wear hats and wear sunscreens. So these are very effective. I'm not sure you know this one or not, but the skin cancer prevention campaign in Australia. So let's watch this video.
Slip, slop, slap, seek and slide, have fun outside, but don't get fried. Slip on a shirt, slop on sunscreen, slap on a hat, 6 shade, slide on sunnies, simple as that. Slip, slop, slap, seek and slide, have fun outside, but don't get fried. Slip, slop, slap, seek and slide.
So this is very important campaign saying slip, slop, slap, seek and slide is important for skin cancer prevention. And what is the impact of this campaign on public health. So these graphs show the melanoma incidence rates in United States and Australia. In United States, melanoma incidence rates are increasing. In contrast, in Australia, SunSmart campaign started in 1987, and since then, incidence rate is decreasing, is slowing down, and then now decreasing. So this is a big impact on public health. So public awareness of skin cancer prevention has a huge impact. So of course, sun avoidance and sunscreens are very effective, but we also want to know how to prevent skin cancer by chemical compound, chemoprevention, especially by using dietary compounds. So can your diet help prevent skin cancer? Yes. So growing evidence shows that the foods high in certain nutrients may indeed help. So this is a pioneer study showing the association between coffee and skin cancer. And this paper said no statistical significant positive associations were found between coffee consumption disease. So this said coffee will not cause disease. That's great. And a strong negative association with coffee drinking were noted for cancer of the kidney and non-melanoma skin cancer. So skin cancer and kidney cancer can be prevented by coffee drinking. So after this pioneer paper in 2000, this paper showed an association between tea and skin cancer. So in this case, hot black tea consumption was associated with reduced risk of developing skin cancer. And moreover, in 2007, now caffeine coffee intake is associated with decreased risk of skin cancer. So in this study, each daily cup of caffeine coffee is associated with 5% reduced risk. And this is dose dependent. So that means that people who drink 6 cups per day showed a 30% reduced risk. And importantly, no risk reduction was found among a decaffeinated coffee drinker. And then risk reduction was less pronounced for tea drinkers, probably due to half caffeine content in tea compared to coffee. So after this paper, more and more papers are published showing the association between caffeine coffee and other types of skin cancer. So not only non-melanoma skin cancer, but also melanoma and especially basal cell carcinoma can be prevented by caffeine coffee intake. And the coffee can also prevent the other types of cancer like hepatocellular cancer and endometrial cancer. And coffee can prevent oral cancer. So more significant effects. So oral cancer, 1 or more cups per day showed a 65% reduced risk of developing oral cancer. And then finally, this paper showing an association between coffee and mortality. So you can see all causes of death here. And then dots are on the left of this middle line, indicating coffee is protective. So coffee intake is associated with decreased mortality in men and women. So decreased mortality in men and women. So this is a great public health impact. So here's another question. So which one do you like? Do you like coffee, black tea, green tea, colas, energy drink, chocolate, or none of the above? Yeah, thank you for joining this poll. Yeah, many people enjoy coffee and many people like chocolate and also black tea. Right, thank you very much. So I think coffee and chocolate and black tea are top 3 choice. This slide shows the caffeine content. So how much caffeine is found in different products. So coffee, 1 cup of coffee may have a 90 milligram of caffeine, and the black tea roughly half caffeine content compared to coffee, and chocolate, so about 20 milligram of caffeine. So if you don't drink coffee or tea, you can eat a chocolate to increase your caffeine concentration. And eating chocolate have another benefit. So this study said chocolate consumption is associated with the number of Nobel laureates per 10 million population. So this is a statistically significant association. This provocative study was published in top journal, New England Journal of Medicine. And I just want to say that this is association, not causation. And this does not say causal relationship, but this is very interesting study. Then how many skin cancers can be prevented by caffeine in the US? So we use a publicly available data to calculate this number. Decreased risk of non-melanoma skin cancer by each daily cup of coffee is 5%, and annual incidence of non-melanoma skin cancer is 5.4 million, and about 50% of US population drink coffee. Among coffee drinkers, average daily coffee intake is 3 cups. Then we estimate that 400,000 non-melanoma skin cancer can be prevented annually in the US by drinking caffeine coffee. So drinking coffee already have a huge public health impact. So I'd like to talk about the science, how caffeine can prevent skin cancer. Our long-term collaborator, Dr. Alan Coney, performed pioneer mouse studies showing caffeine suppresses UV carcinogenesis. So in his mouse studies, mice were irradiated with UV for 40 weeks. And then this graph shows the number of squamous cell carcinoma per mouse, so how many skin cancers developed in this mouse. So compared to water vehicle control, green tea drinking mice showed less tumor development, but the decafeinated green tea didn't. But the caffeine alone or decafeinated green tea plus caffeine also reduced the skin cancer development after chronic UV exposure. So this study clearly said that caffeine is an important compound to prevent the skin cancer. So how does caffeine prevent skin cancer? Caffeine augments UV-induced apoptosis in mouse skin. Apoptosis is one form of cell death, and the caffeine application augments UV-induced cell death compared to acetone vehicle control. So what does this mean? So augmentation of UV-induced apoptosis or cell death may be beneficial to eliminate DNA damaged cells that are precancerous so that the skin cancer development will be suppressed. So caffeine is a non-specific small molecule that affects many protein and enzymes. So we wanted to know what is the actual target of caffeine to prevent the skin cancer. And the caffeine also inhibits ATR. So we hypothesize that the ATR may be the target of caffeine to prevent the skin cancer. So we wanted to see in vivo effect of ATR inhibition on UV carcinogenesis. So we generated a mouse model in which ATR kinase is functionally inhibited. In this mouse, TG, UV-induced apoptosis was augmented. And how about the tumor development? So tumor development was significantly suppressed in this mouse after chronic UV radiation. So again, UV-induced apoptosis was augmented when ATR kinase is inhibited. And also ATR inhibition suppressed UV carcinogenesis, indicating ATR may be a caffeine's target for skin cancer prevention. Here's a model. If ATR is active in cells, UV generates DNA damage and induces apoptosis and cell death, but this apoptosis may be limited. Some DNA damaged cells remain and skin cancer may develop. In contrast, if ATR is inhibited, UV-induced apoptosis may be augmented. So this augmentation of cell death may be beneficial to eliminate DNA damaged cells so that the skin cancer will be inhibited. So these studies show that ATR inhibition prevents skin carcinogenesis, but we did not address this question. What is the optimal timing for caffeine or ATR inhibition to suppress UV carcinogenesis? UV-induced skin carcinogenesis is a long-term process, and the caffeine may be effective immediately after UV or long after UV. So we wanted to know when caffeine should be present to effectively prevent skin cancer. Here's our experiment design. In delayed caffeine schedule, mice were first irradiated with UV for 14 weeks. So at the end of 14th week, mice were at high risk of developing skin cancer, and those mice were treated with caffeine or vehicle cream. In immediate caffeine schedule, mice were irradiated with UV, and after each UV irradiation, caffeine or vehicle cream was applied. So how was the tumor development? So this graph showed the percent squamous cell carcinoma-free mice. So when tumor is developed, this curve is going down. In delayed caffeine schedule, no difference between caffeine and vehicle group. In contrast, immediate caffeine significantly inhibited the tumorigenesis. So caffeine was effective only when caffeine was applied immediately after UV. So effect only seen immediately after UV. So translational impact of this finding could be coffee at the beach could further prevent skin cancer, or caffeine in sunscreen. So caffeine needs to be present at the time of UV, not long after UV, to effectively prevent skin cancer. So why is immediate caffeine effective for skin cancer prevention? A better understanding of ATR kinase activation mechanism will help to answer the question. So we performed molecular studies to elucidate how cells sense UV-induced DNA damage. So again, UV generates DNA lesions and activate ATR check1 signaling pathway in cells. But UV generates these two major types of lesions. One is a cyclobutane pyrimidine dimer, CPD. Other one is a 6,4 photoproduct, 6,4-PP. Both are dimers formed by UV, but these two types are structurally distinct. Compared to CPD, 6,4-PP is highly DNA distorting, but less abundant. And then 6,4-PPs are rapidly repaired by nucleotide excision repair, and it's highly mutagenic if unrepaired. So we wanted to know which type of DNA lesions, CPD or 6-pp, is responsible for AT activation. To dissect the respective effects of CPD and 6-pp, a problem is that the UV generates two types of lesions simultaneously. So we cannot know which one actually induces ATR activation. To solve this issue, so we use a lesion-specific photolyase that can remove either CPD or 6-pp upon light illumination. Photolyase is a unique enzyme that is not present in humans. Photolyase is present in plants and marsupials. So we introduced the photolyase into human cells to generate the cells with a single type of lesion. So now we can know which type of lesion may activate ATI check1 pathway. This graph shows the phosphorylation of check1 as an indicator for ATR activation. And the first three samples are sham no UV control, so no ATR activation. Fourth bar showed ATR activation in cells with both type of lesions. How about in cells with a single type of lesion? Cells with 6-pp activated ATR, but not in cells with CPD. So this is a striking difference. So only 6-pp activates ATR. And this is surprising because 6-pp is a much less abundant type, so 8-fold less abundant compared to CPD. So here's our model. UV generates CPD and 6-pp, and CPD is a more abundant type, but no effect on AT activation. 6-pp blocks DNA replication and activates ATR. So what is the role of ATR activation? So this may promote lesion bypass process. So ATR may promote lesion bypass to survive UV damage, but at the same time, this may be a problem because the mutations will be incorporated and the number of mutations will increase. So ATR is good for survival, but the mutation will be incorporated. So if this was the case, ATR inhibition should be a good thing because that will prevent the skin cancer. And indeed, we performed a genetic study showing good ATR inhibition prevent the UV carcinogenesis, and also immediate caffeine prevented the UV carcinogenesis. So in conclusion, UV generates so many DNA lesions. So remember, 1 hour of sunlight generates 100,000 DNA lesions per cell, which are potentially mutagenic. However, most of the lesions are repaired by efficient DNA repair mechanism. So we are lucky because we have a nice DNA repair mechanism in human cells. So enjoy the sunshine, but not too much. Need to think about these public health campaigns, sleep, stop, suck, sick, and slide. So too much sun is a problem. And also enjoy coffee, especially at the beach. Each daily cup of coffee is associated with 5% reduced risk of skin cancer. And the caffeine also prevents other types of cancer. So enjoy coffee, especially at the beach. But not too much, because I don't, so don't drink 20 cups of coffee per day to reduce the skin cancer risk to 0. So that is too much. And also see a dermatologist. So prevention and early detection of cancer is so important. So see a dermatologist if you have a question about your skin changes. Or become a dermatologist. I really encourage you to join a medical field. So this medicine is a very exciting field where we can discover disease mechanism, develop new therapies, and care for patients and families. I would like to thank my lab members and my mentors and collaborators and their funding sources. To learn more about our research on photobiology and skin cancer, please visit our website at kawasamilab.org. And then finally, I'd like to get your evaluation. So please scan this QR code to submit your evaluation. So thank you very much for attention, and I'm happy to take any questions. So I also put the link for evaluation in the chat. So I appreciate your feedback. So if you have any questions, please type in the chat and I'm going to answer from the top.
All right. Excellent presentation. We do have a couple questions. I'm just kind of scrolling down. Sorry. Let's see. Okay, one of the questions is, what is the risk reduction if you add milk to coffee or tea?
Risk reduction comparing your tea and coffee?
Risk reduction if you add milk.
Or add milk?
Mm-hmm.
A great question. So in my opinion, caffeine content is important. So adding milk may not have additional benefit or disadvantage. So caffeine content is related to skin cancer prevention. So no effect from milk.
Milk, okay. Next question, can you speak to the topical application of caffeine a little bit more? And thank you, I found the study you cited very interesting.
Great question, thank you. So topical application of caffeine was performed in mice, and we wanted to know the direct benefit of applying a caffeine cream onto back skin of mice. So we also performed the oral administration of caffeine into mice, and then we see a similar effect. So oral caffeine and topical application caffeine both prevented UV-induced skin carcinogenesis in a mouse model, and then no clear difference between topical application and the oral caffeine. I think both are effective, but if you apply caffeine-containing cream onto your skin before UV exposure, there may be additional benefit because caffeine absorbs UV energy. So if you apply caffeine-containing cream before UV energy will be absorbed by caffeine. So that means less number of DNA lesions can be generated. So that is an additional sunscreen effect of topical caffeine.
Next question, thank you. Do you have a brand of recommended caffeinated sunscreen?
That's a great question. So I don't have a particular product to recommend. I think the important thing is that the SPF of 30 or higher to prevent your skin from UVB, and also broad spectrum to prevent your skin from UVA as well. So UVA and UVB are different wavelengths, and UVA will cause tanning and skin aging, and the UVB is more responsible for sunburn and skin cancer. But preventing from both UVA and B are important. So broad spectrum water resistant sunscreen is recommended.
Great. Next question, how often should a fair-skinned person be examined by a dermatologist for cancer screening, and does coffee drinking affect this?
That's a great question. I think an annual checkup may be nice for early detection. And you can also examine your skin by yourself. So self-examination is highly effective. And what was the last question?
Does coffee drinking affect the screening?
Coffee drinking effect on screening. That's a great question. I think coffee, theoretically, coffee drinking will reduce the risk of skin cancer, but we don't know your sun exposure history. So some people are more exposed to sunlight, like outside workers. So that will increase the risk of skin cancer development. So I think a balance between outdoor activities versus coffee consumption. So I think it's nice to talk with your dermatologist to see how often your skin should be examined.
Okay, next we have a couple more questions. Does the temperature of the caffeine make any difference in skin cancer prevention?
That's a great question. I think some studies want to measure caffeine content in different brewing time or temperature, and then coffee bean types. So I think it really depends on your cup of coffee. So actually brewing time, temperature, coffee bean all affects the caffeine content in your cup of coffee, and then caffeine content will be different. So that is an important point, but we believe that the caffeine content is most relevant to skin cancer prevention, and that will be affected by many factors, including the brewing and temperature.
Great. Has there been any human studies conducted with topical caffeine?
As far as I know, there's no human studies for caffeine containing cream. But actually, some skincare products already contain caffeine. So some people may already benefited from using those products. But there's no formal study for caffeine in human.
Great. Well, as you can see in the chat, people definitely thanked you and loved the presentation. Looks like we'll have time for one more question. Lots of thank you. That was fantastic. Last one is, what would be an effective dose of caffeine in cream?
Effective dose. So is that talking about the topical application?
Yeah. So in the sunscreen, where you can see like how many of what, I think they're asking like, what would the number or amount of caffeine? That's
a great question. I don't remember how much caffeine we put in cream for mouse studies, but I think a similar number as oral caffeine is highly effective. So if you think about your daily cup of coffee, caffeine concentration is 2 millimolar. So by using the number, you can also calculate how much caffeine you want to put in sunscreen.
Great. Yes, definitely, Chris. Fascinating topic. Thank you so much